So a little background about us. Goldfinch Bio was launched roughly 2 years ago by Third Rock Ventures. We're located in Cambridge, Massachusetts. 30 full-time employees and growing. And my role in the company is that I work in informatics and IT as part of the computational research group. Unfortunately, our headcount hasn't grown nearly as quickly as our AWS bill, so we're only at 4 people, 3 PhD scientists and myself. So a little bit about the company. So our mission is to lead a renaissance in the treatment of kidney disease. And kidney disease is a global epidemic affecting 1 in 9 people worldwide. And staggering amount of money is spent on it, almost 20% of the US Medicare budget. And unfortunately, the field really hasn't progressed in the last 50 years since the advent of dialysis and transplant. There really haven't been any new treatments for proteinuric kidney disease in that time. But there are a lot of companies working in this space, obviously. We're fairly new, only 2 years old. But using recent technology advances and personalized medicine, we think that we have a good chance to kind of make a breakthrough here. So the company's product engine is driven by 2 core components, the Kidney Genome Atlas, which will be the focus of the rest of the talk, and then the biology platform. And so we're focused on our personalized medicine approach in that we're finding new targets to drug and also finding patients to drug. And so in doing that, we're taking 2 parallel analysis paths downstream. So we have our focused analysis where we have a set of known genes that are involved in kidney function, and we look at variants from those genes, and we test them in an experimental assay. And then we also have an unbiased analysis where we're doing things like a genome-wide association study, identifying variants, and then, again, funneling into our biology platform where we can use Hugen organoids derived from patients with kidney disease, and that's what's shown there in a microscopy image. So the Kidney Genome Atlas is essentially a large multi-omics data set of patients with kidney disease. At the core of it is whole genome sequencing data. We've started in FSGS, which is a rare form of kidney disease, and we've sequenced over 2,000 patients there, in addition to several thousand patients from the reference population, which are healthy patients without kidney disease. In addition to that, we also have some RNA-seq data where we are able to look at transcriptomics from the same patients that we have sequenced, and then also looking at biopsies, immunohistochemistry, some proteomics data, and then we have longitudinal medical records on all of these patients, going back as far as we can get them from their medical providers. And then also looking at some newer sequencing technologies like single-cell RNA-seq. And our goal is pretty ambitious. Over the next couple of years, we're hoping to sequence an additional 25,000 patients moving into the diabetic nephropathy space, as well as sequencing 25,000 patients from the reference population, which are patients with diabetes but without kidney disease. So to try to tie all this multi-omic data together, initially we're building a genotype phenotype correlation kind of interface, which is a web-based interface written in AngularJS, using APIs built in the API gateway with Lambda functions written in Python. And at the core of that is a query builder where any of our clinical scientists and translational scientists can go in and define a cohort of patients of interest based on demographic data, lab results, any of the phenotypic data we're collecting. And then we present to them kind of population-level visualizations like Kaplan-Meier plots, box plots, things like that, that show the overall population of the cohort that they've defined. And then they're able to drill down into that cohort to look at patient-level details in individual patients, where we have kind of a per-patient page where they can look at all the data we've collected from any of those patients over time, and also a predicted molecular diagnosis, with the hope that they can then start looking at commonalities between these patients and start digging into what are the common genotypes related to that phenotype that we're looking at. Supporting this interface is a large amount of relational data, so we're using Amazon Aurora and a whole suite of relational databases that we've built. So with any large-scale sequencing project, we have multiple collaborators, we have multiple sequencing CROs, and so just keeping track of the logistics of all that data is pretty challenging. And so we have databases and APIs to help us with all of this. Samples are constantly failing. We have multiple batches going on at the same time across different providers, and a lot of metadata associated with that we keep track of. And then we also have a lot of QC metrics, so before samples get sequenced with library prep, they get tested to make sure the DNA is good quality. And then during the pipeline data analysis as well, we collect a lot of metrics there that we dump back into our relational databases. And then we have longitudinal medical records, so clinical data, medication data over time. And that comes in a variety of formats. Every data delivery we get from every collaborator is a different format. And the data needs a lot of cleaning, kind of massaging, before we can load it into a normalized schema. Then we also have quite a bit of reference data. There's other multi-omic data. There's public GWAS data, gene expression data, and proteomics data, and then annotations as well from public databases, gene variant annotations. We use the Nomad database from Daniel MacArthur's lab at the Broad. And then in the first half of next year, we're hoping to add a clinical data warehouse to this as well as we begin our first clinical trial, where we're enrolling patients from the KGA that we already have a lot of data on, and we'll hopefully be able to add those data points as well to what we already know about them. All of this data is versioned over time because it's really important that all of our results are reproducible, so all the schema has baked into it kind of versioning tables and immutable records. So we're running a standard DNA-seq workflow. When the company launched, we decided very early on that we needed to start with a commercial vendor to do the orchestration of the sequence analysis because we thought we just didn't have the resources or the bandwidth with a very limited staff to be able to do that ourselves. So we partnered with a commercial organization that could do the orchestration, and we decided we wanted to use the GATK workflow out of the Broad Institute as kind of the gold standard in this space. And so we licensed Sention, which is a commercial implementation of GATK that comes with support, which was very appealing to us. And we worked with that commercial vendor to implement the GATK workflow, or the Sention workflow, on AWS for us. And we ran into a lot of issues with that approach. It was very buggy, didn't really deliver as promised. And so we realized pretty quickly that this wasn't a good scalable solution for us long term. So now we're in the process of re-engineering that natively on batch. We've been playing with Cromwell out of the Broad Institute as a workflow orchestration engine. And the folks at AWS did a nice job adding support to that with batch. And then all of our data is just stored on S3. We're pretty aggressive about archiving that to Glacier. And then we are doing, to your question earlier, we're doing joint calling on a quarterly basis across the entire data set. And so the way that we do that is we split the genome up into roughly 1,000 segments. And then for each of our 6,000 samples, we push roughly 6 million files on that data set to S3. And then we orchestrate this on batch. It's basically just a big MapReduce problem where we map over all the segments. We apply the joint calling algorithm. So we get roughly 1,000 VCFs as a result, and we merge those together. And then we do variant quality score recalibration, where it's essentially MapReduce and the other dimension where we're splitting on samples instead of genomic regions. So we get our final output there, which and all of this data lives on S3. And then we also have a QC pipeline as well that gets orchestrated as part of that as well. So as I mentioned with our commercial solution, we had a lot of problems with scalability. They also have a data warehousing component, which was really a data silo. And as we're building out this interface and this multi-omics data set, that just really wasn't a good scalable solution for us. And so we're in the process now of re-architecting that as a native data lake, basically in S3. So we'll take our view QSR data, and we'll load that onto an EMR cluster right now. I'm pushing that in HDFS. And then using the Atom libraries, we convert the VCF to Parquet. And that enables us to use AWS Glue to create an external table in the Metastore, and then use Athena to query that data. And once we're able to write basic SQL queries against it, we can start doing some basic filtering and looking at variants in genes that are significantly enriched in the kidney disease population versus the reference population. And we also use the Nomad database to select for variants that are significantly more rare compared to the reference population. We've also been using Amazon QuickSight for basic dashboards about the project. We have so many different cohorts and so many different batches of data being sequenced at the same time. We needed some easy visuals to kind of understand what the status of each of those batches are and each of the different cohorts are. And then we're hoping to be able to deliver to our collaborators as well an interface where as they're recontacting patients, they can actually update that in real time through the web interface, and we'll have additional dashboards related to that. So we have a very patient-centric kind of view of the data set that we're building, as well as the view of growth over time of the data. We're also using EMR quite a bit for our downstream analyses, as I mentioned, with the focused and unbiased analysis. And so we're using HAIL from the Broad Institute as our genomic framework that we're doing our analysis on, which runs on Spark. And so we're able to bootstrap an EMR cluster with all of that installed. We also adapted the VEP libraries from the Broad Institute to be able to run on Amazon for variant annotation, which has been really helpful. And so there we're able to use Spark and a reasonably large cluster to do our genome-wide association studies. We've also been looking at eQTL analysis. And then these downstream analyses kind of funnel towards our ability to identify new targets as we layer on additional information, public research and literature and things like that that we add on as well. And then with the introduction of urinary proteomics, we're able to also look for biomarkers. We've also been doing network analysis in the data set, and so shown on the left is just a Kaplan-Meier plot of predicting kidney survival over time. But we're also been playing with loading the whole network of the whole human genome into Amazon Neptune, which is a graph database, so that we can start asking questions like what's the neighborhood around a gene of interest, what are the edge weights between different genes that we might be interested in, and hopefully deliver something like this animation I have up here, which is a force-directed graph written in D3, where we could present that to a user where they can look at the topology of the graph in a region of interest and start understanding kind of what are the genes, what are the neighbors around genes that are interesting. And then we can use that to expand our focused analysis so that we have a larger set of genes to start with that we're interested in. So in summary, we've been able to do a lot with a really small team in a fairly short amount of time, thanks in large part to AWS and the platform that they offer. And hopefully, relatively soon, we'll be able to bring new medicines to this underserved population.